Hello, everyone. I hope you're all doing well today. Now, today we're going to look at ETC and chemiosmosis. We're going to look at the steps involved, and also at the end of the lesson, I'm going to give you some summarized notes that you may use to prepare for exam. All right, and so before I get into the processes and the steps, I'm going to look at some background information. And so we all need energy to carry out our life processes. So we do need energy to carry out all different types of work within inside our body. And so in our bodies, our cells are responsible to carry out respiration. And that is why the process is called cellular respiration. However, though, within the cells, there are some specialized organelles called mitochondria that actually is responsible for respiration. And that is why the mitochondria, they are called the powerhouse of the cell. However, though, before we can talk about ETC, we should know that ETC is a final stage of cellular respiration. And so there are two other phases that take place before ETC. One of them is called glycolysis, which is the first stage of respiration. It takes place in the cytoplasm, which is outside of the mitochondria. Glycolysis is responsible to produce H plus ions and also NADH. And these two substances or molecules are very important for ETC. The second phase of respiration is called Krebs cycle. And the Krebs cycle takes place in the matrix of the mitochondria. Krebs cycle produces FADH2 and NADH as well as H plus ions. Now, these molecules will move into the inner membrane of the mitochondria where ETC will take place. A very important point to note right here is that there are thousands of ETC occurring within the inner membrane of the mitochondria. So it's not just one ETC, there are thousands occurring simultaneously. And because you're producing a whole lot of energy at the same time all over your bodies. All right, so let's look at the mitochondria in a little bit more details just to understand exactly where ETC is taking place. Again, on the outside of the mitochondria, we have our cytoplasm. And the cytoplasm, as you know, is a jelly-like substance that keep these organelles in place within the cell. Now, the mitochondria is a double membrane organelle. I'm going to look at that shortly. Now, the innermost part of the mitochondria is the matrix. And then now we have our two membranes. We have the very outermost one, which is called the outer membrane. And then the innermost membrane is called the inner membrane. What is important, between these two membranes, we have what they call the intermembrane space. And so we need to know these membrane for the process that we're going to look at right now. Okay? So let's look at the membranes to understand how ETC actually takes place. Because remember, we said ETC takes place within the inner membrane of the mitochondria. Now, looking into the membrane, again, just to notice on the very outside of the membrane or the mitochondria, we have our cytoplasm. So the cytoplasm is on the extreme part or the very outermost part of the mitochondria. Then we have our outer membrane, then we have our intermembrane space, then we have our inner membrane space, and then the matrix, which is the central part or the most central part of the mitochondria. Now, what is important here, just to point out this real quick, is that the two processes we mentioned earlier that we're focused on today, they are ETC and we have our chemiosmosis. Now, important to note right here is that ETC is where oxidation takes place. And for chemiosmosis is where phosphorylation takes place. And that is why you see most times you refer to ETC as a oxidative phosphorylation process. And I'm going to explain oxidation as we go along and also phosphorylation. Now, for ETC to take place, which is the first process, as you see on the diagram right here, is that it involves four important proteins. And they are protein complexes, as a matter of fact. And so we have our protein complex 1, our protein complex 2, our protein complex 3, and our protein complex 4. Now, along with these protein complexes, we have some very important proteins as well that assist with the process, and they are pretty much cytochrome proteins. The first one is a coenzyme, and is a coenzyme Q. And the other one that assists with this process that comes after protein complex 3 is cytochrome C. Now, for chemiosmosis, very important protein that is involved right here, and this protein is called ATP synthase. And by the name, you know that this is an enzymatic protein because it ends with ASE. So it is an enzymatic protein. All right, let's now dive into what is taking place in the intermembrane. Again, just point this out. You notice that the ETC takes place within the inner membrane of the mitochondria. So notice these proteins that we're highlighting, they are along the inner membrane of the mitochondria. All right, so the first thing that happened here, let's consider it this way, is that coming from glycolysis and the Krebs cycle, again, we have some important substances, NADH and FADH2, along with H plus ions. Okay, so please remember that from our two previous processes. And so here in protein complex 1, our NADH will be oxidized into NAD plus. And oxidation takes place here because it is a loss of hydrogen. So a loss of hydrogen or electrons is called oxidation. And so once this oxidation reaction takes place, electrons will be released and also H plus ions. Okay, what is going to happen here now to these two molecules, which is electrons that are released from NADH and the H plus ions, is that the electrons will move down the inner membrane into coenzyme Q. The H plus ions will move into the intermembrane space. All right, and remember there are a lot of NADH coming from both previous processes. All right, and so you could go back to glycolysis process that I've done and also the Krebs cycle. You will see the number of NADH that actually produced by those two processes. Now, the next thing that will happen here that is very important as well is that FADH2 that is produced in the Krebs cycle, it will also be oxidized, but it will be oxidized by protein complex 2. And once it oxidizes, it will produce FADH. Remember again, the loss of hydrogen. So again, electrons will be produced and also hydrogen ions will also be produced. However, unlike protein complex 1, the H plus ions will not move across the inner membrane into the intermembrane space. They will stay into the matrix, and that's very important. The electrons that are produced will move into coenzyme Q. From coenzyme Q, the electrons coming from protein complex 1 and 2 will move down into protein complex 3. From protein complex 3, what will happen here is that some H plus ions now that were released from the protein complex 2, they will move into the intermembrane space at this time, and electrons now will be sent into the cytochrome C. Now, electrons from cytochrome C will move into protein complex 4. Now, what is very important here is that protein complex 4 will carry out a very important process. All right, so let's look at the process that is taking place here now. And is that H plus ions will also move over into the intermembrane space, because remember, there are a lot of H plus ions within the matrix of the mitochondria, just to make a note of that. But what will happen here is that the electrons that move into protein complex 4 will be used to power the reaction between H plus ions and oxygen to produce water. And that's very important that is taking place there. The water, again, is a byproduct of respiration. Now, because of the movement of H plus ions into the intermembrane space, what you have here is a high concentration of H plus ion in the intermembrane space. And because of this, you will create a gradient of these hydrogen ions, a difference in concentration, high and low concentration. So at this point, the matrix will be low in hydrogen, and in the intermembrane space will be high in hydrogen ions. So what will take place here, just like osmosis, this is now chemiosmosis, the movement of substances from high to low concentration, but in this case is chemicals, so it'll be the ions that will be moving. But it's the ATP synthase that will be responsible to take these H plus ions from the intermembrane space back into the matrix. So they will move down that gradient. And so once the H plus ions are moving down, the H plus ions will help to power the ATP synthase and cause a turning effect. From this turning effect, now what will happen here is that ADP, which is adenosine diphosphate, will be phosphorylated. So the phosphorylation of ADP will take place by adding a phosphate group. So notice the word phosphorylation is adding a phosphate group, and is adding to ADP to turn into ATP. So adenosine diphosphate, which is 2-phosphate group, added as another phosphate group, becomes adenosine triphosphate, which is now 3 phosphate groups. And thus now we finish with the 2 processes of ETC and chemiosmosis. A point to note, there's a lot of ATP will be produced in the ETC process and chemiosmosis. Now, let's jump into summarized notes, and you can use these notes to guide you in terms of preparation for examination. And so first thing I want to look at here is that ATP is produced in 2 ways. One way is that it is substrate level phosphorylation. And this occurs during glycolysis and also the Krebs cycle. And it involves kinase enzymes to produce small amount of ATP. So during glycolysis and Krebs cycle, you only produce a small amount of ATP. The other way that ATP is produced is by oxidative phosphorylation, and this occurred during chemiosmosis, where NADH and FADH2 that we just looked at, they lose hydrogen by the process of what? Oxidation. And then the H plus ions power the formation of ATP from ADP by phosphorylation, producing large amounts of ATP. And that's why we say that the ETC, along with chemiosmosis, is called oxidative phosphorylation because of oxidation of the NADH and FADH2, and then the phosphorylation of ADP into ATP. All right, some more notes right here, real quick. Again, the electron transport chain, just to give you a little brief definition for that. And the ETC is a pathway involving 4 specialized proteins. We call them the protein complexes from 1, 2, 3, and 4. And they send electrons down the inner membrane of the mitochondria. The purpose for the ETC is to increase the concentration of H plus ions within the intermembrane space. And additionally, within the matrix, H plus ions and oxygens are converted into water, and that is done by protein complex 4. Some more notes again is that in chemiosmosis, short definition for chemiosmosis is pretty much the movement of ions, and in this case is H plus ions, or what we call protons, along a concentration gradient across a semi-permeable membrane. And in this case for ETC, it is through the ATP synthase. Now, the purpose of chemiosmosis is to use H plus ions to energize the phosphorylation of ADP into ATP. Now, our overall process is that we have protein complex 1 and 2, again, where oxidation takes place, and then the electrons move into coenzyme Q, then into protein complex 3, and then into cytochrome C, and then into protein complex 4, and this is what they call ETC. Now, after ETC is completed, then we move into our chemiosmosis, where we have a lot of ATP being produced. Now, importantly right here is that protein complex 1, I'm going to give you just very brief notes on the protein complexes. So protein complex 1 contains NADH dehydrogenase. And very important to note this because notice the word dehydrogenase because it is removing hydrogen from NADH. And that is the oxidation process right there. All right, so it oxidizes NADH into NAD plus, and it pumps now the H plus ions from the matrix into the intermembrane space, and it pushes electrons along the inner membrane into coenzyme Q. Now at protein complex 2, what takes place right here is that it also contains what they call succinate dehydrogenase, and in this case, the succinate dehydrogenase will remove the hydrogen from the FADH2. So it oxidizes FADH2 to FAD. It also pushes electrons along the inner membrane into coenzyme Q. And very important to note right here that protein complex 2 does not pump H plus ions across the membrane. The H plus ions, they remain in the matrix at this point. Now, importantly is that coenzyme Q, otherwise called ubiquinone, and right here, just to make a note right here, is that it is mobile. It is a mobile electron carrier. Now, it transports electrons to protein complex 3. Now, going into protein complex 3, now it pumps 4 H plus ions from the matrix into the intermembrane space. It pushes electrons along the inner membrane into cytochrome C. Now, for cytochrome C, a very important protein right here, it transports electrons into protein complex 4. And very importantly, at protein complex 4 is that it pumps 2 H plus ions from the matrix into the intermembrane space. It uses electrons to energize the formation of water. Oxygen at this point will split into half, so you only have one oxygen, and it combines with 2 H plus ions to form H2O. So notice there are 2 atoms of hydrogen and 1 atom of oxygen within water. So split the O2 into half, producing only 1 oxygen that combines with 2 H plus ions to form water. All right, so very important right here, again, just to make a note, is that the ATP synthase, all right, and what happened at ATP synthase is that we have high concentration of H plus ions in intermembrane space, and so they will move through the ATP synthase into the matrix, so it pumps H plus ions into the matrix. While doing this, the ATP synthase catalyzes the phosphorylation of ADP into ATP, and phosphorylation is adding a phosphate group to ADP. All right, and just remember that a lot of ATP is produced. As a matter of fact, the most ATP is produced at the ETC and chemiosmosis process. All right, so we're at the end of the lesson right now. I just want to tell you that I truly appreciate you watching, and I want you to know that we all have something great inside of us, and let no one tell you otherwise. So at this time, keep safe, be blessed, have a wonderful day. See you soon.